Hi everyone, welcome to today's biology practical session. So today we are going to study about common disease causing organisms. So where this will be a question number 5 in your exam. This question carries 2 marks. 1 mark will be for identification of the organism. So the organism we may provide in the form of a slide, or it may be a specimen, or it may be a chart. So you have to identify the organism and you have to name the disease caused by it. So identification carries 1 mark and the disease caused by it carries 1 mark. Now to start with, so let us know what is disease. So disease literally means that the state of discomfort. So if we feel some discomfortness or uneasiness, so that may be due to the entry of some pathogen or a parasite where we call it as a disease. So disease means the state of discomfort. So the disease may be of 2 types, infectious and non-infectious. So we here study about infectious disease. Infectious disease, you all know that it spreads. It spreads from person to person by various type of agents, through air, through water, through vectors like mosquitoes, housefly and etc. So infectious disease, mainly focusing on it in the present experiment. So if I say infectious agent, the organism which is able to spread from one person to another person, and it is having the potency to induce some changes in our body that may lead to some discomfort, which we finally call it as a disease. And every disease is characterized by its own symptoms. Now for example, if we have an infection by some bacteria, that is through food, which has infected the intestine. Now related to that, we may show the symptoms like diarrhea, abdominal pain and uneasiness, loss of appetite. So these are some symptoms which is specific to that particular bacteria which has entered the intestine through food or water. So specific organism shows a specific kind of symptoms. Now by knowing the morphology and the life cycle of a particular disease causing organism, it is more helpful to study the disease as well as to treat the disease. And very important thing, the symptoms. The symptoms will be the first sign for us to identify the type of organisms. So based on the symptoms only, the diagnosis will be done further. So symptoms are very important to identify the type of disease and in turn the type of pathogen. So once we identify the pathogen, it is very easy for us to treat the disease. So knowing about disease causing organisms and the symptoms they show, it's very important and it is helpful in treating the diseases. So today's experiment we are going to study about 4 different type of disease causing organisms. And you all know that the disease causing organisms are called pathogens. So we are going to study about the pathogens and the disease caused by them and the related symptoms. Yes, now to start on with, we will start with the organism entamoeba. So you all know that amoeba or entamoeba, it belongs to the phylum Protozoa. It belongs to the class Rhizopoda, and the type, the scientific name of the organism is Entamoeba histolytica. So it's very important to note this point. Once you identify the organism by seeing a slide or by seeing a chart or by seeing the specimen, it's must and should you have to write its scientific name. So you all know the rules of binomial nomenclature, but still I again repeat. So you have to write both genus name as well as the species name. Genus name should start with capital letter, species name should start with small letter, and both the words should be underlined separately. Now once you write the scientific name of the organism, you will get 1 mark. Then, now once we come to the ant amoeba, this is actually one of the stage of ant amoeba life cycle. So this stage is called trophozoite stage, and this is infectious stage, and in this stage we could diagnose the ant amoeba. So I can call it as it is the diagnostic stage of the pathogen ant amoeba. And ant amoeba is an endoparasite. Endoparasite. Endo means inside. Parasite means which lives inside the body of the host. So endoparasite. So ant amoeba lives inside the body of human. Actually it lives in the intestine of human. Now as I told, this is one of the stage of ant amoeba life cycle where we can diagnose ant amoeba at this stage. So it's very important to know the morphology of this stage. Once we see the structure, so make sure that it is microscopic and it is acellular. Microscopic acellular form. What does it contain? First main thing is nucleus. So if you observe here, the nucleus is single and it is accentrically located. Accentrically means away from the center. Mostly we observe that nucleus will be almost present in the center of the cell. But here we are noticing that the nucleus is away from the center, so that is accentrically placed single spherical nucleus. Now once I observe within the nucleus there is a dark spot. Here I am showing that there is a dark spot. The dark spot is called as karyosome. The karyosome is covered by a layer of chromatin. So it's not that chromatin is organized in the center of the nucleus. Here the chromatin forms a single layer, peripheral layer. So in the center there is karyosome. Surrounding the karyosome there is a single layer of nucleoprotein. Nucleoprotein is nothing but chromatin. And the chromatin is surrounded by nuclear membrane. Now I repeat again, nucleus is single, accentrically placed. It has a central dark spot called karyosome that is surrounded by a single layer of chromatin granules, and chromatin granule is enveloped by nuclear membrane. So you will observe this nucleus within the ant amoeba. Then here we are observing many structures, granulated structures. So these are food vacuoles. So there are more number of food vacuoles present inside the ant amoeba at its trophozoite stage. And there are no contractile vacuoles present in the ant amoeba. So this is about the structure of ant amoeba, where you have to identify it by seeing the slide, and the clue for it will be the darkly stained nucleus. So you can keep a darkly stained nucleus as a clue for identifying the ant amoeba at its trophozoite stage. So once you see the slide, you will identify the ant amoeba, and then you have to report two things. One, the name of the organism. As I mentioned earlier, you are going to write the scientific name where you follow the rules of binomial nomenclature. That carries 1 mark. And the second thing what you are going to report is the disease caused by it. The disease caused by it is amoebiasis, or it could also be called as amoebic dysentery. So if you write the name of the organism and the disease caused by it, you will be scoring 2 marks. Okay, next we will study about the second organism, that is Plasmodium. So you all know that Plasmodium causes malarial disease in humans. Plasmodium belongs to the phylum Protozoa, class Sporozoa, and the scientific name is Plasmodium vivax. So Plasmodium, as I told before, it causes malarial disease in humans. So it is a digenic intracellular parasite. So digenic, di means two. So it requires two hosts to complete its life cycle. The primary host is female Anopheles mosquito and the secondary host is human. So that's why we say that female Anopheles mosquito is a vector for the Plasmodium to reach the human. So through the mosquito bite, the Plasmodium will enter the human body, that means into the bloodstream, at its infectious stage called sporozoid. So sporozoid is the infectious stage of Plasmodium. At that stage, the Plasmodium makes its entry into the human bloodstream through the mosquito bite. Now it's very important to diagnose the disease. Now a person is suffering from malaria. He will be noticing some symptoms like intermittent high fever, and he may have chills, and he will have a severe muscular pain, loss of appetite, sleeplessness. Mainly all these are basic symptoms of malaria. He will go to diagnosis where they will take the blood sample and they will diagnose it. So very important thing is, we can diagnose the malaria at a particular stage, the stage called Signet ring stage. And I mentioned earlier, the Plasmodium is intracellular. It will be present inside the cell of the host. Here the particular target for the malarial parasite is RBC, red blood cells. Now we can observe the parasite inside the RBCs in the form of a ring. This ring is called Signet ring stage. So if a person is suffering from malaria, we can test the person for the particular disease at this particular stage. So we call it as the diagnostic stage of malarial parasite is Signet ring stage. So here if you observe, this is a parasite and here there is a central hollow space. This is actually the food vacuole, which is large. So the large food vacuole pushes the cytoplasm to the periphery where it forms a ring. And we can observe that the nucleus is pushed towards one end and the nucleus seems dark in color. So this overall structure appears like a Signet ring and hence it is called Signet ring stage, which we observe inside the human RBCs. So once you observe the slide for Plasmodium, you have to notice this Signet ring within the RBC. So once you observe a ring structure within RBC, then you will identify it as Plasmodium vivax. Again I repeat, identification carries 1 mark and the disease caused by it, that is malaria. If you write this, you will get 1 mark. So totally you can score 2 marks. Coming to the third organism, that is Ascaris. So you have studied this organism in detail in your first year. So as you all know, Ascaris belong to roundworms, that is the phylum Ascihelminthes. It belongs to class Nematoda and the scientific name is Ascaris lumbricoides. Now commonly called Ascaris is roundworm. The very important feature or the significant feature of Ascaris is that it shows sexual dimorphism. That means we can clearly distinguish males from females. So here this is a male Ascaris which is short and its posterior end is coiled and it is thin. So male ascaris is short and thin compared to female ascaris, and its posterior end will be coiled up. Then coming to the female ascaris, it is thick and long, and its posterior end is not curved like male ascaris. So we can clearly, by seeing, we can clearly differentiate between a male ascaris and a female ascaris. And structurally, this is the anterior end. Anterior end bears the mouth at its extreme tip, and below the mouth towards the ventral side there is a excretory pore, aperture called excretory pore. And in females, if we come down 1/3 from the anterior end, we are able to see the genital pore. Then the body, in both male and female, it has dorsal and ventral surface, and there are two lateral lines that demarcate the ventral and the dorsal surfaces. So this is a lateral line which we observe. Then once we take a male ascaris, at its cloaca, the opening, there are a pair pineal setae. So this pineal setae will help during copulation. Then the female ascaris, as I mentioned earlier, it has a genital pore at the 1/3 position from the anterior end. So now by knowing the morphology of this ascaris, it's very important to know that it's an endoparasite. It lives inside the body of the host. So here the host is human being. So it lives in the intestine of the human. And the symptoms what we observe once there is infection by ascaris is, we observe a severe abdominal pain, anemia, fatigue, and there will be occasional vomiting, and this is also characterized by diarrhea. So by knowing these symptoms, it could be predicted that the infection is by the ascaris. So once you write the ascaris by identifying, so ascaris usually we will provide it in the form of a specimen, a bottle. Once you identify it, if you write the scientific name, you will be getting 1 mark, and the disease caused by it, that is ascariasis. The disease caused is ascariasis. So once you mention the disease, you will be getting 1 mark. So totally it carries 2 marks. Okay, now coming to the 4th one. Actually this is the last one in the present experiment, that is trichophyton. So trichophyton is a fungi. It belongs to kingdom Mycota, and it belongs to the class Ascomycetes fungi. The name of the organism is Trichophyton rubrum. This is actually ectoparasite, and mostly you all know that fungi will feed on the dead biomass. So that means it is on the human body, it feeds on the skin. Mainly it causes skin infections. So here the disease caused by this organism is ringworm disease. And this disease, as I told, it's basically a skin infection. The disease or the infection area is most commonly the scalp, and it is the region between the fingers, and it's the region between the toes where there is moisture. So you all know that fungal organisms, they require moisture to grow. So they prefer the areas like between the regions between the fingers, the region between the toes and the scalp, where they get the moisture and they grow. And once we see the symptoms of this infection, there will be itching, intense itching in the region of infection, and there will be a ring. The ring is surrounded by a fluid-filled lesions, and hence we call the disease as ringworm disease. So this disease, as I mentioned earlier, basically caused due to the growth of fungi, that is Trichophyton rubrum. And since it infects the skin, it's called a ectoparasite. Once we see the structure of this fungi, so usually we provide this organism in the form of a chart, where the chart will also have the same diagram. It's very easy for you to identify. You will see the hyphae. You know that the fungal body, the growth of the fungi, what we observe on the substratum, that is hyphae. And this hyphae bears the reproductive structures called macroconidia and small microconidia. If you notice this hyphae with macroconidia and microconidia, you are going to report it as Trichophyton rubrum, and the disease caused by it is ringworm disease. So identification carries 1 mark, the disease caused by it carries 1 mark. So altogether you will be scoring 2 marks. So among these 4 what I have explained, you may get any one organism in your exam. So you may have a slide, or you may have a chart, or you may have a specimen for identification, and the question will be among these 4. Any one you may get. So your work is to just identify, write its scientific name and write the disease caused by it. Okay, now I'll be telling you the clue or the hint to identify each of these specimen in your exams. To start with entamoeba. Entamoeba, as I mentioned earlier, it is microscopic. So we have to use the slide to identify it. And the clue for identifying entamoeba is the darkly stained nucleus. So now in a slide, if you observe a oval shaped cell within which there is a darkly stained nucleus, you can report it as entamoeba histolytica. Then coming to the plasmodium. Plasmodium, the very important stage is the signet ring stage. So this will be the chart which will be kept in your exam. So this signet ring, if you notice, then identify it as Plasmodium vivax. Then coming to Ascaris. Ascaris will be the specimen, the bottle. So you can observe the unsegmented long round worms where you will identify it as Ascaris. Then coming to Trichophyton. So Trichophyton, as I written the diagram on the board, it is same the chart. So it's very easy to identify the Trichophyton by looking at the hyphae, macroconidia, microconidia. If you see this, you will be identifying it as Trichophyton rubrum.